Hi everyone, welcome back to my video presentations. Here we will discuss RNA processing or post-transcriptional modification of RNA. So we know that after transcription, the RNA molecules which are synthesized are designed to perform various functions. Let it be the template for translation, or it may be a structural feature, a structural function as seen in tRNA or rRNA molecule. But whatever be the function, in order to be functional, these molecules has to be subjected for certain treatments. And these treatments include some additions, some deletions, some rejoinings, and collectively all these treatments are called RNA processing or the post-transcriptional modifications. So these modifications are very much essential for the RNA molecule to be functional. So let's see what are the different mechanisms which are performed usually in the RNA molecule. The molecule after synthesis, we call them as the primary transcript. So these primary transcript usually undergoing an extensive processing in eukaryotic system compared to a prokaryotic cell. The three most important RNA processing mechanisms are splicing, 5' capping and polyadenylation. Splicing is a mechanism to remove introns. So what are introns? Introns are nothing but the non-coding regions within the mRNA molecule. So the counterpart, that means the coding regions within the mRNA, are called exons. So there will be exons and introns seen in the mRNA molecule. So of which we need to remove the introns and rejoin the exons together. That process is called splicing. And the second and third one, that means the 5' capping, is usually the addition of a nucleotide, a special nucleotide, into the 5' end, and polyadenylation is the addition of adenine residues to the 3' end. And all these mechanisms together usually will work out to produce a functional mRNA molecule. So the capping usually provides some protection to the 5' end of the mRNA molecule from endonuclease activities. So as already we know that these mechanisms are very much evident in eukaryotic system compared to prokaryotes. Even though we can see slight mechanisms of RNA processing in prokaryotes, especially in tRNAs and rRNAs, but in all the different forms of RNA, in eukaryotes we can see mostly at least one of the processing mechanism. So this is the outline of RNA processing mechanisms. In the 5' capping, a number of enzymes and proteins are involved, collectively called them as cap synthesizing complex. And in the polyadenylation process, we have an important enzyme called Pol Il polymerase. This is nothing but just like a DNA polymerase enzyme, but the thing is that which doesn't require a template. It can add adenine residues to the 3' end. And the splicing we will discuss a little detail in the next section of the video, where we have different types of introns like type 1 to type 4. And all these 4 different types of introns are processed by different mechanisms. Like the type 1 and type 2 are performing self-splicing, whereas the type 3 will be mediating a spliceosome mechanism, and type 4 is an endonuclease mediated mechanism. And what are be the types, what are be the type of RNA processing? Most of the proteins associated with the RNA processing are usually seen attached to the polymerase enzyme itself, that means to the RNA polymerase enzyme. usually to the carboxy terminal domain of the RNA polymerase enzyme. So this RNA synthesis and RNA processing are closely related. So this shows you how this mechanism is executed. You can see the mRNA molecule with the exon sequences and the intron sequences. The yellow colored ones are the intron sequences. They has to be removed, and through the cleavage, that means the splicing mechanism, you can see the introns gets removed and the exons are joined together. And at the 5' end, there is an addition of a 5' cap, and at the 3' end, the adenine residues are added. And now we are moving into the mechanism of polyadenylation process. So as already said, there is addition of around 80 to 250 nucleotides to the 3' end. And we know that the addition of these adenine residues occur at the 3' end. So during the termination process of transcription, we know that the cleavage occurs at a particular sequence, and this cleavage site is demarcated usually around 10 to 30 nucleotides downstream to a particular sequence called AALAAA. So the cleavage occurs at that site. That means usually 30 nucleotides downstream of the AALAAA site. And for the cleavage, actually this sequence is a marking for polyadenylation also. And there are many number of proteins are attaching onto this site, which include the CPSF, CSTF, cleavage factors, and along with all those proteins, there is a significant protein called Pol I polymerase also. And this Pol I polymerase is responsible for the addition of adenine residues after cleavage by the cleavage factor, which already we have discussed in the termination mechanism of transcription. So this Pol I polymerase will add adenine to the already cleaved 3' end of the RNA molecule. You can see that mechanism in little more details. The CPSF, that means the cleavage and polyadenylation specific factor, will recognize the AALAAA sequence, and usually 10 to 30 nucleotides downstream of this binding sequence, there will be cleavage, and this cleavage is facilitated by the cleavage factors. Usually cleavage factor 1 and 2 will be acting together to perform the cleavage. And after cleavage, there are many other proteins also involved. The CSTF is one example, the Pol I polymerase is another one, and these Pol I polymerase will cause the addition of adenine residues at the 3' end. And just after the addition of Pol A tail at the 3' end, there are some stabilizing protein called Pol A binding proteins, and these Pol A binding proteins will come and bind onto this Pol A tail, and these are required for stabilization, and not only for stabilization but also these Pol A binding proteins are required for the successful initiation of translation. Right, so the second mechanism called capping at the 5' end. This is nothing but addition of a 7-methyl guanosine residue to the 5' end of the mRNA molecule, and this occurs just after the initiation of transcription. That means at around 20 to 30 nucleotides have been synthesized from the transcription machinery, the 5' end will be freely available, and at that time itself the capping will be done. And this capping usually occurs through a multi-step process, and you can see the structure of the 7-methyl guanosine. This is nothing but a modified base of guanine. There is addition of a methyl group at the 7th position, and these 7-methyl guanosine will form a special unusual bonding with the 5' end of the mRNA. You can see the first and the second nucleotides of the mRNA at the 5' end, and to the last phosphate group, there you can see they will be having usually 3 phosphate groups will be there in the first nucleotide of the mRNA molecule, and to that phosphate groups there will be a special bonding, commonly we call it as the 5' triphosphate linkage, and through that linkage these 7-methyl guanosine will be attached into the 5' end. You can see the mechanism in detail. There is the last nucleotide which is shown with all the 3 phosphate groups, and the gamma phosphate group will be released by an enzymatic activity of phosphohydrolase, and then there is addition of an external guanine into the system, and this guanine will be transferred into the mRNA through catalysis of guanylyl transferase enzyme, and there is cleavage of 2 phosphate groups, and the remaining adenosine monophosphate will attach to the 5' end, and you can see they attach in that fashion. And this is the addition of the 7-methyl guanosine, and actually this is addition of the guanosine, and after this addition of guanine residue to the 5' end, there is a methylation process, and this methylation is done by the enzyme guanine 7-methyl transferase, and the methyl group is supplied by S-adenosine methionine or adomate, and there is addition of the methyl group into the 7th position. So that's about the addition of 7-methyl guanosine into the 5' cap, and occasionally there will be addition of methyl group to that already existing nucleotide bases in the mRNA. That means to the second position of the first nucleotide of the mRNA at the 5' end also will be done, and that's done by another enzyme called 2' methyl transferase enzyme. So after synthesis, usually as already said, these proteins and complexes are usually associated with the carboxy terminal domain, and after the synthesis of the cap or after the successful addition of the cap, usually this complex, that means the proteins responsible for the addition of the cap will be released out, and this cap soon after the synthesis of the or addition of the cap will be bound by another set of proteins called cap binding complexes, and which are usually involved in the interaction of these 5' cap to the translation machineries. So this is 5' capping process. So I hope you understood this polyadenylation and 5' capping. You get an idea on that, and in the next video we will see the third and most elaborated processing called splicing. So discuss these things and have your queries. Okay, thank you for watching.